In the shaded undergrowth of a warm forest, where leaf litter muffles footfalls and every shadow holds its own private conflict, a solitary wasp drifts just above the ground. Its flight path looks casual, almost lazy, but this is the deceptive calm of a surgeon moving toward the operating table. Below, a cockroach rummages through decomposing leaves, oblivious to the precision instrument descending upon it. The air between predator and prey is quiet, but beneath that stillness pulses one of the strangest strategies in the insect world. A parasitic hijacking so precise it might as well be considered neurological surgery. The wasp, slender and metallic in the light, is equipped not merely with a stinger, but with a biological syringe capable of delivering complex chemical instructions. Many insects use venom to kill, immobilize, or defend, but parasitic wasps have pushed this concept further, evolving toxins that act not simply on muscle tissue or pain receptors, but directly on the nervous system. Their venom does not aim to end a life quickly. It aims to rewrite it. The cockroach senses movement and flares its spiny legs, preparing to run. Speed is the roach's first and most reliable defense, but the wasp accounts for this. It dives with a burst of acceleration, lands expertly on the roach's back, and immediately curls its abdomen under the body. The first sting is fast. It pierces the soft membrane between the roach's armored plates and injects a measured dose of paralyzing venom into the thoracic ganglia. This structure acts much like a spinal generator, controlling movement and reflexive escape responses. In a healthy roach, the nerves fire rapidly, driving the instinctive sprint that makes the species notoriously hard to catch. But once the wasp's venom seeps into the tissue, those signals fade. The legs remain intact, the muscles still viable, but the neural command center goes eerily silent. What follows is one of nature's most unsettling pauses. The cockroach remains upright, its antennae twitching, its legs splayed in what should be a scrambling escape. Yet it cannot run. The paralysis is temporary and localized, tailored carefully so the wasp can move to the next phase, direct access to the brain. This is where the operation becomes so exact that entomologists have compared it to neurosurgery performed without tools. The wasp circles the roach's head, searching for the tiny gap at the base of the antenna where the neural sheath is exposed. Here lies the subesophageal ganglion, a region responsible for integrating signals related to motivation and voluntary movement. The wasp aligns its stinger, which is tipped with sensory structures that allow it to feel its way through soft tissue, and makes the second injection. This venom behaves very differently from the first. It is not meant to paralyze. Instead, it interferes with neurotransmitters that govern the roach's sense of agency. It dampens the pathways that trigger avoidance, exploration, or escape, leaving intact the basic circuits that control walking, limb coordination, and equilibrium. The result is a creature that is not dead, not unconscious, and not physically restrained, yet no longer capable of making decisions. The roach can walk perfectly, it can climb, it can navigate obstacles, but it cannot initiate a single one of these actions on its own. The wasp has effectively unplugged the brain's decision-making center, leaving behind a functioning body waiting for external input. To test whether the procedure has succeeded, the wasp performs a subtle behavioral check. It gently backs away and waits. A healthy cockroach, even after a moment of shock, would attempt to flee, but this one remains still. The wasp approaches again and taps one of the roach's antennae with its jaws. The roach responds with a small step, not self-initiated, but triggered by the tactile stimulus. With that, the wasp grabs the antenna like the handle of a leash and begins to walk. The roach follows. This is perhaps the most haunting scene in the forest undergrowth. A predator leading a fully conscious prey animal as though it were trained livestock. The roach walks with steady coordination, descending through soil channels and over moss-covered stones, moving wherever the wasp guides it. There is no struggle, no attempt at escape, no hint of awareness that its behavioral autonomy has been replaced by a chemical override. The wasp drags the roach into a small burrow it has prepared earlier. Deep underground, the temperature remains stable, humidity high, and the threat of scavengers minimal. This is not a hunting cache. This is an incubator. Once they reach the chamber, the wasp climbs onto the roach's body and delivers a final sting, not to the nerves this time, but to the outer tissue. This does not kill the roach. Instead, the wasp deposits a single egg against its body. The roach sits quietly throughout the process. Its breathing is steady. Its legs hover in place, alive yet purposeless. After securing the egg, the wasp exits the burrow and seals the entrance lightly. Its work is done. The roach, still alive and fully capable of movement, but unable to choose to move, remains inside the chamber. Within a couple of days, the egg hatches into a larva. It climbs onto the roach and begins feeding. In one of nature's most efficient and unsettling survival strategies, the larva eats selectively, avoiding vital organs to keep its host alive for as long as possible. As the larva grows, it consumes more tissue, yet the roach persists, sustained by the careful design of the wasp's venom that suppressed panic responses and movement but spared the metabolic systems needed to keep the host alive. Evolution has sculpted this strategy to remarkable precision. The venom does not merely paralyze or sedate, it modulates neurotransmitters such as octopamine, a chemical similar in function to adrenaline. By suppressing octopamine-driven pathways, the wasp dampens the roach's capacity to interpret danger or initiate escape. Experiments have shown that even strong stimuli, such as a puff of air that normally sends cockroaches sprinting, produce little more than a mild twitch in a venom-altered roach. The insect retains full muscular control, yet behaves as though the impulse to flee has been erased. This manipulation offers immense evolutionary advantages. By keeping the roach alive and immobile, the wasp ensures that its offspring have access to fresh, uncontaminated prey that will not decompose prematurely or attract competing scavengers. A dead roach decays quickly, dries out, or becomes infested with microbes. A living roach remains a stable, protein-rich environment, warmed by its own metabolism and defended by its intact immune system. In this sense, the wasp larvae benefit from a fully maintained biological shelter, one that remains operational until the moment they're ready to break free. Inside the chamber, days pass in silence. The larva grows fat on the roach's internal tissues. Eventually, when it has consumed enough to fuel metamorphosis, it delivers the final bite, killing the host. The larva then spins a cocoon within the same burrow and transforms, emerging as an adult wasp, ready to repeat the same chilling ritual. This cycle showcases an evolutionary path defined not by brute force, but by neural manipulation. Instead of overpowering prey, the wasp hijacks its behavior. Instead of killing quickly, it preserves life just long enough. The wasp's venom acts as brain malware, infiltrating the neural network, disabling critical functions and leaving the rest untouched. It is one of the few natural examples of targeted neurological control, practiced by an animal with no formal brain science, only the accumulated wisdom of millions of years of evolutionary refinement. In the quiet corners of the forest, far from human eyes, this drama unfolds countless times. Yet each instance remains a testament to the complexity of natural selection and the surprising strategies organisms evolve to survive. The wasp is not simply a hunter, it is a technician, a manipulator, and in the evolutionary sense, an engineer of behavior. Its venom is not a weapon, but an instrument, one that performs brain surgery with the precision of a needle and the finality of instinct. Morning light settles across a meadow, sliding over dew-beaded grasses and drifting between wildflowers that sway under the faintest breeze. Beneath this peaceful surface lies a meticulously arranged hunting ground, held together by silken lines so thin they catch the light only when the sun strikes from the perfect angle. Suspended between two tall blades of grass, the web of the wasp spider glitters like a tensioned instrument, its radial threads converging on a carefully engineered spiral. The spider itself occupies the center, motionless except for the slow pulse of its abdomen. It is a creature of vivid warning, wrapped in bands of yellow, black, and white that resemble the bright stripes of a stinging insect. This mimicry is not accidental. It is a message broadcast across the meadow, "Do not interfere." Yet the spider's true violence unfolds not in stings or bites, but in a quieter and more methodical form of execution. A grasshopper vaults upward from the base of the meadow, its legs extending with a burst of stored elastic energy. It clears several centimeters in a single jump, landing directly onto the web. The impact reverberates through the strands like a plucked string. Immediately, the grasshopper's legs flail, trying to gain purchase, but the silk clings to their spines, stretching just enough to trap motion without snapping. The sticky globules along the spiral capture each limb with adhesive precision, pulling the insect deeper into the trap with every frantic move. The spider senses the tremors within a fraction of a second. It does not rush blindly. Instead, it orients itself toward the source of vibration, reading the frequency and rhythm as though decoding a signal. This allows the spider to distinguish between wind, debris, and living prey. Confident in its interpretation, it moves with controlled speed, crossing its own web without disturbing the pattern. Each step is placed on non-sticky radial threads that function like structural beams. The spider approaches the writhing grasshopper, its legs unfurling in a slow, deliberate arc that suggests both caution and mastery. The first contact comes when the spider raises its front legs and positions its fangs near the grasshopper's thorax. The grasshopper still has strength, its hind legs kicking with enough force to send predators flying. But here, the silk absorbs the energy, anchoring the insect in place. The spider leans forward, and its fangs pierce the exoskeleton exactly where the armor is thinnest. Venom flows instantly, a cocktail designed not for rapid kill, but for systematic shutdown. Inside the grasshopper's body, the venom binds to neuromuscular junctions. It prevents the transmission of signals that trigger muscle contraction, leading to a progressive failure of movement. Instead of an immediate collapse, the grasshopper's limbs enter a strange sequence, twitching, then shuddering, then growing still. The muscles stop responding, but the sensory system remains partly aware, registering the gradual decline in control. The insect's compound eyes continue to reflect the world around it, though it can no longer act upon what it sees. This paralysis is not dramatic or bloody. It is quiet, clinical, and inevitable. As the venom spreads, the spider retreats slightly, giving the immobilization time to deepen. The grasshopper's wing muscles, normally capable of rapid, powerful contractions, fall silent first. Then the legs stiffen. The abdomen pulses irregularly as internal rhythms falter. When the spider returns, the prey is alive but defenseless, its body held against the web like an offering. This is when the true process begins, the part that rarely appears in casual observations of meadow life. The spider positions itself again and sinks its fangs more deeply this time, not merely injecting venom, but releasing digestive enzymes into the grasshopper's tissues. In spiders, digestion happens outside the body, an adaptation that bypasses the limitations of a narrow feeding tube. The enzymes act like a liquid scalpel, breaking down complex biological structures into fluid form. The chemistry is astonishing. Proteins unravel into short peptides and individual amino acids. Cellular membranes weaken and rupture, releasing their contents. Organs soften, losing their structural integrity as the enzymes permeate deeper layers. Hemolymph, the insect equivalent of blood, begins to mix with the dissolving tissues, forming a thickening slurry. This transformation happens internally, hidden behind the intact cuticle of the grasshopper, giving the process an eerie cleanliness. There is no open wound, no spilled viscera, no visible disassembly. Instead, the prey appears almost peaceful, as though merely resting in the web, while inside its body, liquefaction has already begun. The spider waits, monitoring the progress with subtle movements of its palps. Over time, the grasshopper's internal pressure shifts, showing the first signs of collapse. The exoskeleton retains its shape, but the structure beneath softens, turning from solid tissue into nutrient-rich broth. This is the spider's external stomach at work, reducing the prey into a form that can be consumed through the narrow mouthparts. When enough internal dissolution has occurred, the spider anchors itself and begins to feed. It places its mouth over the entry point created by the fangs and draws in the liquefied tissues. The feeding is slow, steady, and efficient. Each sip removes more internal mass, leaving behind a hollow shell that still looks from the outside like an intact grasshopper. Over the course of minutes or sometimes hours, depending on the size of the prey, the spider converts a once powerful jumper into simple nutritional paste. What makes this process so striking is the contrast between the spider's appearance and its method of killing. The wasp spider's patterns are vivid and beautiful. The yellow stripes mimic warning colors found in stinging wasps, discouraging many predators from attempting an attack. Yet the method of predation remains hidden behind controlled movements and a lack of overt gore. The death it offers is slow and internal, a biological dissolution rather than a physical tear. It is a refined brutality, quiet but thorough, shaped by the demands of a predator that must conserve energy and avoid unnecessary risk. From an evolutionary standpoint, this approach is remarkably efficient. By immobilizing but not immediately killing its prey, the spider ensures the body remains fresh during digestion. Dead insects degrade quickly, attracting bacteria and losing moisture. Living but paralyzed prey maintain internal homeostasis longer, keeping tissues in optimal condition for enzymatic breakdown. This strategy also reduces the risk of injury to the spider. A grasshopper's kick can be lethal to a small predator, but venom-induced paralysis eliminates that threat within moments. The external digestion process itself is a marvel of biochemical adaptation. Spiders secrete a blend of hydrolases, enzymes that target fats, proteins, and carbohydrates, and each compound contributes to the smooth transformation of tissue. Proteases dismantle structural proteins like actin and myosin. Lipases break down fat stores. Nucleuses dissolve genetic material into manageable fragments. The spider consumes the resulting mixture with minimal waste, leaving behind a lightweight husk that barely resembles the struggling insect that entered the web earlier. Around the feeding spider, the meadow continues its routine. Bees drift over flowers, ants navigate stems like miniature highways. Birds call from hidden perches. None of them witness the slow collapse taking place on the web. The grasshopper is now little more than a silhouette of its former self, its body emptied with scientific precision. When the spider finally releases the carcass, the breeze catches the hollow shell and drops it among the grass, where it becomes another small fragment of the meadow's nutrient cycle. The wasp spider resumes its position at the center of the web, motionless once more, waiting for another vibration to carry through the silk. Its stripes gleam in the sunlight, a combination of beauty and warning that hints at the predator within. Yet the true story of its hunting method remains hidden from casual observers. The web may appear delicate and the spider itself elegant, but every detail, from venom to enzymes to the architecture of the silk, serves a purpose honed by countless generations. The quiet execution of prey through chemical transformation. In the humid air of a late tropical afternoon, the forest canopy glows with sunlit fragments. Light filters through gaps in the leaves, scattering across trunks and vines until it settles on one of the most extraordinary structures in the understory, a sprawling golden web, stretched wide like a suspended tapestry. Each strand glimmers faintly, catching even the weakest rays and throwing them back with a metallic warmth. This is the architecture of the golden silk orb-weaver, a spider known not only for the strength and luster of its silk, but also for a venom that strikes the nervous system with stunning precision. The web is both beacon and trap, its beauty contrasting sharply with the violence hidden behind its threads. A beetle, heavy-bodied and iridescent, hums clumsily toward the web. Its wings blur with a low, resonant buzz as it navigates through shafts of sunlight. The golden threads appear invisible until the moment of impact. Then the air seems to tighten around the beetle. Its wings catch in the silk, folding abruptly. The beetle tumbles, hanging upside down as the threads cling to its legs and wing cases. Each attempt to kick free only binds it more securely, the elasticity of the silk allowing the beetle's movements to pull it deeper without breaking the structure. The vibrations travel instantly through the framework, radiating outward in concentric waves. The orb-weaver, resting near the hub of the web, responds with a subtle shift of its long legs. Its body is a study in contrasts, delicate and suspended, yet clearly capable of sudden, decisive action. The spider approaches with a slow, almost measured grace, pausing occasionally as though analyzing the tremors that ripple through the web. These signals are rich with information, revealing the size, strength, and struggles of the trapped insect. When it reaches the beetle, the spider moves cautiously. Beetles, especially hard-shelled ones, can injure predators with strong mandibles or powerful legs. But the orb-weaver is not merely relying on its silk for restraint. It carries within its fangs a biochemical arsenal designed specifically to disarm such threats. The spider lifts its front legs, positions its fangs near the beetle's thorax, and strikes. The puncture is clean and fast, barely perceptible to the eye, but the venom begins its work immediately. Unlike the digestive enzymes used later, this venom is engineered for rapid interference with the nervous system. It does not aim to sedate, it aims to overwhelm. Inside the beetle's body, the venom components target ion channels, microscopic gateways that regulate the flow of charged particles across nerve cell membranes. These channels maintain the delicate balance of electrical signals that allow an insect to move with coordination and purpose. By disrupting this balance, the venom turns the nervous system into a battlefield of chaotic impulses. Almost instantly, the beetle's muscles begin to spasm. Its legs jerk uncontrollably, scraping across the silk in sharp, erratic motions. The wings beat out of sequence, slapping helplessly against the web. Every muscle receives contradictory instructions, as though the nerve commands have fragmented into static. Sodium channels remain open too long, allowing electrical signals to fire continuously. Calcium levels surge. Neurotransmitter release becomes erratic and unregulated. What should be a precise exchange of signals becomes a storm of neurological noise. From the spider's perspective, this storm is a sign of success. The orb-weaver retreats slightly, avoiding the convulsions that ripple through the beetle's body. The spasms continue, each one weaker than the last. The venom is not merely overstimulating the nerves, it is exhausting them. After a prolonged period of uncontrolled firing, the cells lose the ability to reset. Ion gradients collapse. Synaptic communication falters. One by one, the impulses fall silent. The beetle that only moments earlier thrashed violently now hangs motionless, every muscle limp, every nerve quiet. This neurological collapse is not random carnage. It is the product of evolutionary refinement, an adaptation that ensures maximum safety for the spider. A struggling beetle could break a leg, tear a wing, or inflict injury, but a beetle whose nervous system has been shut down presents no threat. The orb-weaver returns once the convulsions cease, walking calmly along the radial lines of its web. It touches the beetle lightly, confirming the absence of resistance. Satisfied, the spider begins the next stage, wrapping the prey in tight spirals of silk. While it binds the beetle, its fangs sometimes deliver additional doses of venom, ensuring complete immobilization. Yet even after the beetle is fully wrapped, the story of the venom continues beneath the surface. Researchers studying orb-weaver venom have uncovered an array of fascinating compounds. Peptides that latch onto specific ion channels, toxins that alter synaptic transmission, and molecules that selectively target insect nervous systems. Many of these components are so selective and efficient that they have become subjects of biomedical research. In controlled doses, some of these venom-derived molecules offer potential benefits to humans. Scientists exploring new treatments for chronic pain, epilepsy, and neurodegenerative conditions have found that the very same toxins that silence insect nerves may provide pathways for modulating human nerve activity. Certain peptides can block pain receptors with remarkable specificity. Others can dampen pathological overexcitation in nerve cells, offering potential relief for neurological disorders. Yet these therapeutic possibilities emerge from compounds that, in the natural world, bring about a rapid and irreversible shutdown of an insect nervous system. Back in the forest, the orb-weaver continues its methodical preparation of the prey. Once wrapped, The beetle hangs like a small cocoon, turning slowly in the breeze. The spider may feed immediately or store the prey for later, depending on environmental conditions and its own energy needs. Though the beetle appears intact on the outside, the work of the venom ensures that its internal physiology has already changed dramatically. Nerve signals remain silent. Muscles are no longer responsive. Metabolic processes drift into stillness. The golden silk orb-weaver itself remains an extraordinary figure, suspended among the radiant threads. Its silk, with a shimmering gold tint, is not merely decorative. The color may help camouflage the web in sunlit woods, making it harder for prey to detect. It may also attract certain insects that mistake the reflective surface for open sky. But the web is only half of the spider's strategy. The other half lies within its venom glands, where chemistry and evolution have converged to produce a tool capable of shutting down a complex biological system with stunning efficiency. For insects, movement depends on rapid-fire signaling between neurons and muscles. This system is beautifully optimized for speed, but its reliance on precise electrical timing makes it vulnerable. The orb-weaver's venom exploits this vulnerability. By altering ion channel behavior, it does not merely interrupt a signal. It creates electrical chaos that quickly becomes unsustainable. This is why the beetle's spasms begin so suddenly and decline so sharply. The body fires every possible signal at once until the system burns out and collapses. It is a controlled destruction, orchestrated to minimize effort for the predator and ensure a predictable outcome. The spider does not need brute strength or prolonged struggle. It relies on the simple truth that even the toughest exoskeleton cannot protect an insect from its own malfunctioning nervous system. As twilight settles over the forest, the golden web takes on a muted glow, shimmering faintly as the last rays of sunlight slip away. The beetle hangs motionless near the center, its internal storm long since extinguished. The orb-weaver remains still nearby, its legs splayed across the web in perfect balance. To a passerby, the scene might look serene. Yet woven into the silk and hidden beneath the beetle's hardened shell is the story of a neurochemical battle, one fought and won in mere moments. Night settles over the desert in a slow exhale, trading blistering heat for a cool, breathless stillness. The sand radiates away the last of the day's warmth, and the wind grows thin, moving across the dunes with the faintest whisper. This is the hour when the black scorpion becomes a shadow in motion. Its armor absorbs moonlight rather than reflecting it, giving the creature the uncanny ability to vanish even when standing in plain sight. It raises its pedipalps, testing the air with sensory hairs that detect vibrations too fine for human perception. The desert is quiet, but not empty. Somewhere between the sparse shrubs and the low rocks, small animals stir, mice, lizards, insects, each carrying its own fragile rhythm of breath and heartbeat. The scorpion moves toward that rhythm with deliberate patience. A mouse noses along the sand, searching for seeds carried by the day's wind. Its tiny footfalls send vibrations across the ground, a steady pattern the scorpion registers instantly. Without warning, the scorpion lunges. Its tail arcs with practiced precision, delivering a single, decisive jab. The stinger punctures the mouse's skin and injects a controlled dose of venom. The mouse squeaks softly, not yet understanding the magnitude of the event. The scorpion retreats a pace, watching. The venom is a chemical cascade, engineered over millions of years to exploit the fundamental architecture of nerve and muscle. It begins at the sting site, entering the bloodstream in a concentrated pulse. The toxins target sodium and potassium channels, microscopic gates that regulate how electrical impulses travel along nerves. In a healthy organism, these channels open and close in strict patterns, allowing muscles to contract and relax in well-coordinated sequences. The venom disrupts this pattern instantly. Sodium channels lock open. Potassium channels fail to reset. Electrical signals fire uncontrollably, sending messages that the body cannot organize or resist. Inside the mouse, the first symptoms are subtle: a twitch in the hind leg, a shudder in the diaphragm, a rapid blinking of the eyes as nerves fire without command. The mouse tries to run, but its stride falters. The scorpion does not pursue. It does not need to. Its venom is already inside the mouse's circulatory highways, riding the bloodstream deeper with every heartbeat. Within moments, the respiratory muscles begin to misfire. The diaphragm contracts too quickly, forcing shallow, frantic breaths. In small animals, breathing is a delicate balance between muscle rhythm and oxygen demand. The venom collapses that balance. As sodium floods nerve cells, the signals that coordinate inhale and exhale transform into chaotic pulses. The mouse breathes faster, then faster still, its chest rising and falling in desperate staccato. The rapid breaths cannot compensate for the decreasing efficiency. The muscles of the chest wall twitch violently, then stiffen in painful contractions. Oxygen levels drop. Carbon dioxide rises. The mouse's body sends emergency signals across its nervous system, but those too become tangled in the venom's interference. The venom spreads upward toward the heart. Here, the constant, reliable contraction of cardiac muscle depends on a precise electrical rhythm. The black scorpion's toxins are well suited to disrupt this rhythm. The heart responds with erratic beats, short bursts, long pauses, then frantic pumping. The mouse wobbles, disoriented. Its pupils dilate as the brain interprets the flood of aberrant signals as a cascade of continuous danger. For an organism this small, respiratory collapse is only a short step away. The diaphragm eventually locks in a rigid contraction, unable to relax. The mouse gasps once, then becomes still, its heart stuttering before slipping into silence. For the scorpion, this slow progression is essential. Its venom is not designed for instant kill, but for controlled immobilization. Quick kills risk overexertion and injury. Gradual shutdown ensures safety. When this venom touches vertebrates larger than a mouse, the effect becomes dramatically different, but no less serious. The toxins still target ion channels, but the larger body mass dilutes the venom's potency. Instead of immediate paralysis, mammals experience waves of intense physiological upheaval. Pain radiates outward from the sting, not because tissue is damaged, but because sensory neurons begin firing non-stop. The brain receives a continuous, overwhelming stream of electrical noise interpreted as burning, stabbing, or pulsating pain. Sweating begins almost immediately as the autonomic nervous system scrambles to respond. Heart rate increases. Blood pressure fluctuates unpredictably. In humans, this reaction can be severe enough to cause dizziness or nausea. As the venom moves through the bloodstream, it affects nerves that regulate internal organs. The lungs respond first. Breathing becomes shallow or rapid. Some individuals feel their throat tightening, not because of swelling, but because the nerves controlling the airway behave erratically. Then the heart reacts. The same mechanism that collapses a mouse's cardiovascular system produces arrhythmias in humans, irregular beats that alternate between too fast and too slow. It is rarely fatal in healthy adults, but the sensation is unmistakable. The heart no longer feels trustworthy. The venom communicates a message deeper than pain. It tells the body that something is catastrophically wrong. The sympathetic nervous system activates fully, saturating the bloodstream with stress hormones. This internal alarm system is meant to help the body flee or fight, but there is no predator to outrun and no injury to heal, only a chemical countdown set in motion beneath the skin. In severe cases, the heart muscles fatigue under the barrage of irregular electrical impulses. The respiratory system can become so strained that each breath feels insufficient. Collapse becomes possible, especially in children or individuals with pre-existing conditions. The experience is not one of quick doom, but of watching the body's internal systems misfire in real time. Scorpion venom earns its fear not through the decisiveness of its kill, but through the clarity with which it dismantles physiology. It is a message written directly into muscle and nerve. Your systems are no longer yours to control. Back in the desert, the black scorpion advances toward the mouse once more. The small body lies still, eyes half open, cooling against the sand. The scorpion grips it with its pedipalps, and begins to eat with a calm, methodical precision. There is no rush. The venom has assured complete safety. The desert wind stirs, pushing thin sand across tracks already fading in the moonlight. In this landscape of extremes, searing heat by day, freezing cold by night, the black scorpion survives by turning biology itself into a weapon. It does not need speed nor brute force. It needs only a carefully measured dose of venom that hijacks ion channels, steals breath, and places the heart on a timer. Evolution has shaped this venom to be both efficient and terrifyingly elegant. It kills small animals swiftly, cripples larger ones in waves of pain and physiological confusion, and reminds every creature it touches that the boundary between life and collapse can be crossed with a single puncture of a curved black stinger. Dawn spreads across the rainforest floor with a hazy, filtered softness, the sun breaking through in thin gold beams that ripple across bark, moss, and littered leaves. The air is thick with moisture, carrying the earthy scent of decomposing foliage and the layered perfume of growing fungi. In this dim, humid world, an army is already awake. Thousands of leafcutter ants pour from their subterranean nest, flowing like a single organism across the forest floor. Their bodies shine with a faint reddish hue, each ant carrying its own precise purpose. Most ants collect leaves. Some guard the foraging columns. Others scout ahead. Then there is the caste prepared for war. Ants with oversized mandibles, thick heads, and venom glands primed for chemical assault. Deep within the leaf litter, a plump caterpillar grazes on a fallen leaf. It moves slowly, its soft body flexing with gentle waves, unaware that the faint vibrations of its chewing have already been detected. Leafcutter ants do not hunt to eat flesh. They harvest plant matter to feed their underground fungal gardens, but they respond to intruders with ruthless efficiency, especially when those intruders threaten their foraging trails or their carbohydrate-rich leaf caches. And sometimes, a soft-bodied insect like this caterpillar becomes an easy source of protein, an opportunity the colony is not willing to ignore. A scout ant reaches the caterpillar first. It pauses, lifting its antennae, assessing the size and potential threat. Within seconds, chemical signals travel outward, summoning reinforcements. Workers and soldiers arrive in a coordinated wave. The caterpillar senses the shifting ground beneath it and tries to inch away, but the ants swarm over it before it has moved more than a few centimeters. Mandibles clamp onto its sides, anchoring the insect to the ground. The caterpillar thrashes, convulsing in waves as dozens of sharp jaws pierce its skin, creating tiny openings along its body. Then, the true assault begins. Ant after ant curls its gaster beneath its abdomen. The stinger is a narrow, needle-like structure, but for leafcutters, the more formidable weapon is the gland just above it, a reservoir filled with formic acid. With a controlled flex, the ants spray the acid in thin, targeted streams directly into the fresh wounds carved by their mandibles. The caterpillar's soft tissue reacts immediately. Formic acid lowers pH, pushing the chemical environment of the wounds into an acidic range that disrupts proteins at the molecular level. Structural proteins like collagen begin to unravel. Cell membranes weaken. Enzymes that require precise chemical conditions lose function entirely. Inside the caterpillar's body, this sudden acidity changes the behavior of ions and intracellular fluids, making it impossible for normal physiological processes to continue. On the outside, the effects are just as damaging. The acid burns through the upper layers of tissue, creating an expanding zone of damage that widens the original bite marks. The caterpillar bucks wildly, its thrashing movements made frantic by the chemical pain that spreads beneath its skin. The ants tighten their grip, holding their positions with mechanical determination. More ants arrive, each adding its own corrosive spray to the growing chemical bath that coats the caterpillar's body. As the acidity intensifies, the caterpillar's muscles begin to fail. Muscle proteins depend on specific pH levels to maintain structure and function. The acid disrupts these delicate bonds, causing fibers to stiffen, cramp, or dissolve. The carefully orchestrated contractions that allow soft-bodied insects to move turn into disorganized spasms. The caterpillar curls and uncoils unpredictably, its body becoming a canvas of chemically induced convulsions. Meanwhile, the ants continue their methodical disassembly. Their mandibles tear at the compromised tissue, pulling free strips of flesh softened by the acidic environment. What would normally be a difficult struggle, trying to cut through the firm, elastic body of a caterpillar, now becomes easier. The chemical assault does not simply harm the prey, it prepares the prey for consumption. The softened proteins are easier to cut, easier to transport, and easier for the colony's fungus gardens to digest once they reach the nest. Inside the wounds, the lowered pH also creates an ideal entry point for microorganisms. Leafcutter ants rely heavily on mutualistic bacteria that live on their exoskeletons, particularly on the cuticle of their thorax. These bacteria produce antimicrobial compounds that help protect the ants' fungus gardens from unwanted pathogens. But in the chaos of attack, the ants' bodies also introduce a variety of environmental microbes into the prey's open wounds. Normally, such microbes would be resisted by an intact immune system. But the acid has already disrupted cellular defenses, ruptured membranes, and stopped metabolic reactions. Infection takes hold within minutes, accelerating the collapse of tissue integrity. This interplay between chemical weaponry and microbial invasion is not an accidental byproduct. It is a powerful component of the ant's ecological strategy. The same formic acid used as an offensive tool is also deployed inside the nest as a disinfectant. Workers routinely spread small droplets across the leaves they harvest, sterilizing the plant matter before offering it to the fungus they cultivate. The fungus, in turn, produces specialized structures rich in nutrients that feed the ant larvae. Without this careful balance, the gardens would succumb to parasitic fungi or bacteria in the warm, humid chambers below ground. Formic acid is both weapon and shield, an elegant solution that protects the colony's food source while simultaneously equipping its warriors with remarkable destructive power. Back at the site of the attack, the caterpillar's movements slow. The chemical burns continue to expand, leaving patches of tissue darkened and liquefied. Small tears in the cuticle leak fluids that mix with the acidic droplets, creating a viscous sheen across the forest floor. The ants do not relent. They climb over and under the prey, biting, tearing, and spraying in coordinated motions. The once smooth body of the caterpillar becomes a patchwork of exposed tissues. Some ants hold firmly with their jaws, anchoring the prey in place, while others systematically dismantle sections of its body. The rainforest around them continues its rhythmic hum, chirping insects, dripping moisture, rustling leaves. But at the scale of the ants, the world has narrowed to this single interaction. This is a battlefield defined not by brute force, but by chemistry. The ants' actions are not random. They are the result of millions of years of evolution shaping a society that relies on sophisticated chemical communication and chemical defense. As the caterpillar's internal structure breaks down, its organs soften into a slurry of partially digested tissue. The acidity disrupts neural firing, causing the final spasms to fade into stillness. The prey is effectively marinated from both outside and within, its body turning pliable under the relentless combination of mechanical tearing and chemical corrosion. When the first major chunk of softened tissue is removed, several workers seize it and begin the journey back to the nest. They navigate along scent trails marked earlier in the morning, ensuring the valuable protein reaches the fungal chambers quickly. The colony thrives on this efficiency. Every piece of tissue torn from the prey contributes to the health of the underground gardens. The fungus digests the organic matter, breaks it down further, and produces nutrient-rich nodules that ant larvae depend upon. The chemical warfare waged above ground directly fuels the biological partnerships below. The ants' reliance on formic acid links predator behavior, agricultural maintenance, and communal survival in one continuous system. By the time the swarm disperses, little remains of the caterpillar. A few scraps cling to the leaves. The forest floor, glistening with residue, quickly absorbs the fluids, and scavengers will finish what the ants have left behind. Within hours, there will be no trace of the struggle, only the quiet continuation of the rainforest's endless cycles. The leafcutter ants resume their march, returning to cut leaves, tend their gardens, and defend their colony with the same chemical proficiency that brought down the caterpillar. Their weaponry is not flashy, nor is it instantaneous. It is precise, corrosive, and deeply effective. Each droplet of formic acid is a message written in chemistry, one that can burn enemy flesh, sterilize garden leaves, and bind the fates of ants, fungi, and entire ecosystems together. Midday in the forest arrives not with brightness, but with a dim, filtered glow. Thick canopies of overlapping leaves break the sunlight into fragments, scattering pale green patterns across trunks and fallen logs. A humid weight presses through the air, carrying the rich scent of tree sap and slow decay. Among the towering trees, everything seems still on the surface. But beneath layers of bark, hidden in the wooden bones of the forest, an army of silent invaders feeds relentlessly. This is the unseen realm of longhorn beetle larvae, creatures that carve their world not in soil or leaf litter, but deep within the heavy, toxin-laced wood of living trees. To a tree, wood is more than structure. It is storage, defense, communication. Many tree species saturate their tissues with complex chemical compounds, phenols, terpenes, alkaloids, and resins, each serving a purpose. Some repel insects, others slow fungal growth. Still others disrupt metabolic processes so severely that most herbivores avoid the bark entirely. The very heartwood, where some longhorn larvae thrive, can contain compounds so concentrated that they burn the mouths of mammals and stunt the growth of competing plants. By chemical design, wood is meant to resist invasion. Yet this is precisely where longhorn beetle larvae flourish. Deep within a trunk, the soft, pale grub chews through the fibers with slow, rhythmic motions. Sawdust-like frass collects behind it, compacting into winding tunnels that snake through the interior of the tree. The larva's body seems almost translucent, its skin stretching over powerful muscles built for biting, pushing, and grinding through cellulose and lignin, materials that few animals can digest. As it tunnels, it encounters chemical defenses that should be lethal. Instead, the larva behaves as though moving through nothing more hostile than moist bread. The toxic compounds that saturate the tissues pass through its body without harm, or in some species, are broken down with such efficiency that the insect can use the nutrients locked inside. This extraordinary resistance begins in the larva's gut. Specialized enzymes line the inner surfaces, each one evolved to dismantle the complex molecules trees rely on for defense. These enzymes are not generic digestive tools. Many species of longhorn beetles possess unique biochemical pathways that break down phenolic compounds, substances known for their antimicrobial and insecticidal properties. Other pathways neutralize terpenoids, transforming them into harmless or even useful molecules. Supporting these enzymes is a microbial community that lives symbiotically within the larva's digestive tract. Bacteria play a central role in converting cellulose into digestible sugars, but they also serve as chemical detoxifiers. Some microbes can bind directly to plant toxins, while others metabolize them into forms the larva can absorb safely. The relationship is so specific that some beetles rely on bacterial strains inherited from their mothers through specialized coatings placed on their eggs. Without these microbes, the larvae would die in tissues their ancestors conquered ages ago. As the larva tunnels deeper, each bite of wood brings new challenges. Resin pockets burst under its mandibles, coating its face with sticky amber. Alkaloid-rich tissues attempt to impair metabolic function, yet the larva keeps moving, its gut chemistry working as an internal shield. The tree responds as best it can, redirecting sap to the wounded area, producing more defensive compounds, and sometimes attempting to compartmentalize the invasion by forming barriers of dense tissue. But these defenses act slowly. The larva acts constantly. Outside, the tree looks healthy. Leaves remain green. Branches sway gently with passing breezes. Only subtle signs betray the intruder. A faint dusting of frass at the base, small exit holes from previous generations, or an unusual number of woodpeckers tapping the bark as they listen for larvae inside. The invasion is quiet, not the work of hordes, but of individual grubs slowly carving chambers and galleries that weaken the tree over seasons or years. What makes this process unsettling is its intimacy. Longhorn beetle larvae do not merely feed on the tree. They live inside its tissues, sheltered by the very structures meant to defend it. They use the tree's toxic arsenal as a resource, thriving where other organisms would die within minutes. And in some species, the larvae absorb traces of those toxins into their own bodies. These compounds accumulate in small amounts, especially in the outer tissues of the larva and later in the adult beetle. The result is a creature that tastes foul, or in rare cases, mildly toxic to predators. Birds that attempt to swallow such larvae may immediately spit them out, discouraged by the bitterness. Some amphibians show signs of nausea after sampling larvae from strongly defended trees. This borrowed chemical protection is an evolutionary twist. The plant's attempt at self-defense becomes part of the insect's shield. Within the tunnels, the larva grows plump over many months. Its body lengthens, its jaws strengthen, and its frass piles into compacted masses behind it. The tunnels serve not only as feeding corridors, but also as sanctuaries from predators. Only animals with specialized adaptations, such as woodpeckers with shock-absorbing skulls and long barbed tongues, can extract larvae hidden within layers of wood. Even parasitic wasps, some of the most skilled insect hunters, struggle to locate larvae inside particularly resinous or chemically defended trees. The same compounds that longhorn beetles digest easily can clog the sensory organs or ovipositors of these predators, limiting their ability to reach the grub. For the forest, the impact of this tunneling can be felt long after the larva matures. The galleries weaken structural integrity, making trees more vulnerable to storms. The hollow spaces become entry points for fungi and other decomposers. Entire branches may die from internal damage, their tissues riddled with winding channels that disrupt nutrient transport. Yet even this destruction plays a role in the forest ecosystem. Standing dead wood becomes habitat for cavity-nesting birds, small mammals, and countless other insects. Fungal spread accelerates the recycling of nutrients within the soil. Longhorn beetle larvae, in a paradoxical way, help maintain the long-term health of the forest by facilitating decomposition and renewal. Still, the individual tree pays the price. As the larva approaches the end of its development, it begins to construct a pupal chamber deep within the heartwood. This space is larger and smoother than its feeding tunnels, lined with compacted frass and wood fibers. Here, the larva transforms. Weeks pass as the soft-bodied grub reshapes into a longhorn beetle adult, complete with elongated antennae that can exceed the length of its body. When the beetle finally emerges, it chews its way out of the trunk, creating a clean, round exit hole. Each hole marks the end of a larva's months-long occupation and the beginning of the adult's search for mates, a new wood where future offspring will continue the cycle. To the casual observer, this hole might seem insignificant, a small round opening among bark patterns. But for the tree, it is the mouth of a labyrinth carved under constant chemical fire. Every tunnel represents a trench dug in defiance of plant defenses, a testament to the larva's ability to neutralize toxins and reshape its environment from within. Longhorn beetle larvae are not simply pests. They are chemical specialists, capable of surviving inside what should be biologically forbidden territory. Their presence is a reminder that the forest's battles are not always loud. Sometimes the most impactful invasions unfold in silence, hidden behind bark and sap, where tiny jaws and unseen enzymes work tirelessly. The nightmare for the tree is not a sudden attack, but a long, slow hollowing, a quiet invasion carried out by creatures immune to poisons designed to stop them. The afternoon forest hums with a quiet tension, the kind that builds when heat presses down on leaves and sap begins to trickle through stems in slow, sticky currents. Under the canopy, moisture clings to every surface. Moss thickens over fallen branches, and the air smells faintly of crushed vegetation and ripening fruit. In this layered world of scent, a small insect moves with deliberate care atop a sunlit leaf. It is brightly patterned in greens, reds, or mottled browns depending on the species, and shaped like a miniature medieval shield. But beneath that charming facade lies a sophisticated chemical arsenal. This is a shieldbug, a creature whose defenses extend far beyond appearances. The insect climbs toward the leaf's edge, pausing occasionally to probe the air with slender antennae. At first glance, it seems harmless, slow-moving, soft-footed, almost timid. Yet the shield-shaped body is not ornamental. It protects a pair of specialized thoracic glands that hold one of the most potent defensive cocktails in the insect world. To many predators, encountering this bug is like stumbling into a microscopic tear gas attack. High above, a small bird flits between branches. Its keen eyes catch the bug's movement, and instinct takes over. The bird drops toward the leaf with practiced speed, wings folding slightly as it aligns its beak with the insect below. Shieldbugs, after all, are the perfect size for a snack, soft enough to swallow easily and slow enough to catch with little effort. The bird lands, hops forward, and thrusts its head toward the exposed bug. The shieldbug reacts instantly. Muscles near the thorax contract, forcing fluid through narrow ducts connected to the gland reservoirs. With a sudden pulse, a fine spray erupts directly toward the predator's face. The bird jerks back, wings beating frantically as droplets hit its eyes and nasal passages. What follows is not a smell one simply endures. The compounds released are chemically reactive, forming a volatile cloud of aldehydes, acids, and assorted organic molecules that irritate mucous membranes on contact. The bird's response is immediate. Its eyes clamp shut, head shaking violently as it tries to clear the burning sensation. The aldehydes attack the delicate tissues lining the beak and throat, triggering an instinctive cough-like motion. The predator retreats, hopping backward with wings partially extended for balance. Only after several seconds does it finally take flight, leaving the leaf and the shieldbug behind. To understand why this reaction is so strong, one must look past the droplets glistening on the leaf surface and peer inside the shieldbug's thorax. The glands occupy a significant portion of the insect's internal architecture. They contain reservoirs of precursor molecules, often amino acid derivatives or metabolic byproducts, that are enzymatically converted into volatile defensive compounds. These molecules are highly reactive, meaning they bind readily with proteins and lipids, especially those in sensitive tissues like eyes and respiratory surfaces. Aldehydes are particularly potent. Their structure allows them to form temporary bonds with biological molecules, disrupting their stability. When inhaled, even in microscopic amounts, they irritate lung tissue by interacting with the lining of air passages. This causes a rapid inflammatory response, forcing predators to gasp, cough, or retreat. For small animals like amphibians or reptiles, which rely heavily on moist membranes for breathing or sensing, this can be debilitating. A lizard hit directly in the face might freeze momentarily, jaw gaping as the compounds overwhelm its sensory receptors. Recovery may take minutes, long enough for the shieldbug to escape, but the effects do not end with vertebrates. For insects, the consequences can be much worse. The waxy cuticle covering most insects acts as a barrier to dehydration and infection. Shieldbug secretions contain reactive lipids and acidic compounds that attack this barrier directly. When sprayed onto a small predator such as an ant or parasitoid wasp, the chemicals dissolve the thin wax layer, destabilizing the cuticle's integrity. Water begins to evaporate rapidly from the affected insect's body. Within minutes, it may lose enough moisture to impair movement. In severe cases, the damage opens pathways for opportunistic microbes to invade, effectively turning the defensive spray into a slow-kill agent. Predators that rely on stealth or endurance become compromised, losing coordination or succumbing to dehydration long after the shieldbug has moved on. These outcomes illustrate how the bug's defense operates on multiple levels. The bright coloration warns larger predators to keep their distance. The sudden burst of chemicals blinds or stuns attackers that get too close. And for smaller enemies, the spray becomes a corrosive film that disrupts biological systems on contact. Each layer of defense works in concert, shaped by evolutionary pressure across countless interactions with birds, insects, reptiles, and even small mammals. Yet the shield bug itself remains deceptively calm through these encounters. Once the predator retreats, the insect slowly uncurls its legs, straightens its antennae, and resumes its path across the leaf. Its body design ensures that the spray is projected outward and away, preventing accidental self-contamination. The gland openings are angled forward or sideways depending on the species, giving the bug the ability to target specific directions. Some species even produce different blends of chemicals depending on the threat. More volatile compounds for flying predators, thicker and more adhesive secretions for crawling attackers. Even within the confines of the forest leaf, the ecological implications of this weaponry extend far beyond the individual bug. Predators learn quickly to avoid shield bugs, associating their distinctive shape and bright hues with discomfort. This reduces predation pressure not only on adults but also on nymphs, which often share similar patterns. As a result, shield bugs can maintain stable populations in environments where many soft-bodied insects suffer heavy losses. Still, the chemical arsenal requires energy to produce. The bug must divert resources from growth and reproduction to fuel its defensive system. This balance demonstrates a central principle of evolutionary ecology. Every adaptation comes with a cost. Yet in the case of shield bugs, the investment pays dividends. Their defense is effective enough that many can forage openly during the day, feeding on sap, seeds, or fruit without relying on speed or camouflage. In some species, the glands evolve further complexity. Certain shield bugs can control not only the direction, but also the quantity of spray, releasing small droplets for minor disturbances and a full burst for serious threats. Others produce compounds with antimicrobial properties, helping protect against bacteria or fungi that might invade wounds or contaminated surfaces. This dual benefit shows how chemical defenses often serve multiple roles, a recurring theme in the insect world, where efficient use of limited resources determines survival. The forest continues its quiet chorus as the shield bug finishes its slow journey across the leaf. Sunlight shifts, dappling the insect's patterned body as it reaches the shaded underside of the branch. There, it settles into a still position, blending into the patchwork of green and brown. The earlier encounter with the bird leaves no visible trace, but the chemical droplets lingering on the leaf surface continue to volatilize, giving the air a faint, sharp odor detectable primarily by creatures sensitive to such cues. The shield bug's defense is not dramatic like a scorpion's sting, or as lethal as the venom of certain spiders. Instead, it is a carefully engineered blend of irritants and corrosives, an airborne barrier that protects the insect through discomfort rather than destruction. Yet this very subtlety makes it effective. A bird that feels its eyes burn learns quickly. A lizard that tastes bitterness and pain develops avoidance. An ant that begins to dry out after contact thinks twice before attacking again. The shield bug survives by turning chemistry into deterrence, shaping its world through invisible warnings rather than brute force. The adorable shield-shaped body hides a miniature factory for chemical warfare. And in the dense, competitive world of the forest, that factory keeps the shield bug alive, one spray at a time. Morning light pours across a cotton field, washing over rows of broad leaves and pale, unopened bowls that swell like small green hearts. The air carries the scent of sun-warmed vegetation and a faint bitterness rising from the plants themselves, a chemical signature designed to repel most herbivores. Cotton defenses are not gentle. The seeds and surrounding tissues store powerful secondary compounds, including gossypol, a molecule potent enough to disrupt nutrient absorption and poison the livers of many animals. In this landscape of toxicity, one insect moves with fearless confidence. Its body is painted in vivid tones of scarlet and black, a contrast so sharp that it almost glows in the morning sun. This is the red cotton bug, a creature that survives and thrives by stealing the very chemical weapons meant to kill it. The bug walks slowly across a cotton boll, probing the surface with its needle-like mouthpart. With a precise motion, it pierces the seed chamber and begins to feed. A droplet of sap, rich in defensive molecules, beads around the puncture. Inside the plant, compounds flow steadily into the insect's body. To many animals, these molecules would cause gastrointestinal distress, neurological disorientation, or organ damage. But the red cotton bug is not an ordinary consumer. It has evolved a digestive system capable of handling these toxins with remarkable efficiency. As the sap enters the bug's body, enzymes in the gut break it down selectively. Some molecules are neutralized and used as nutrients. Others pass through nearly unchanged, traveling into specialized tissues where they accumulate. Instead of eliminating these chemicals, the bug stores them deliberately. This process, known as kleptotoxicity, turns the insect into a walking reservoir of borrowed plant defenses. The red and black warning colors advertise this fact to the world, signaling danger in a language well understood across ecosystems. Nearby, a young lizard hunts along the leaf litter. Its keen eyes detect movement on the cotton plant, and instinct draws it upward. The red cotton bug is exposed, moving slowly, seemingly easy prey. The lizard creeps closer, flattening its body against the stalk, then strikes with rapid precision. Its jaws clamp around the bug, and for a brief moment, everything appears routine. Just another insect becoming part of the food chain. But almost immediately, the lizard hesitates. Its body stiffens. A convulsive shake ripples through its head. The bug's internal chemical cargo has entered the lizard's mouth. Some compounds irritate the mucous membranes of the mouth and throat. Others begin to act systemically as the lizard swallows. Toxins originally designed to deter mammals and insects now disrupt the reptile's physiology. Mild neurotoxins interfere with chemical signaling. Bitter compounds trigger nausea. Certain molecules cause the liver to activate emergency detoxification pathways, diverting metabolic resources and slowing normal functions. The lizard drops the bug, shaking its head in discomfort. A second attempt seems unlikely. The association between bright colors and unpleasant consequences is formed almost instantly. The red cotton bug tumbles to the ground but lands upright, its legs already moving as though nothing has happened. The plant's own defenses now surround and protect the insect, carried within its tissues as an invisible yet potent shield. This strategy reflects a powerful evolutionary logic. Plants invest energy in creating chemical barriers to protect their seeds. Insects like the red cotton bug hijack these compounds, absorbing and repurposing them for their own survival. The plant's poison becomes the insect's armor. This layered interaction illustrates how chemistry flows through ecosystems, shaping the relationships between species in ways that are both subtle and profound. Feeding continues throughout the day. Each bug selects seeds or sap reservoirs according to its needs, accumulating toxins in varying concentrations. The process is slow but continuous. Over time, an individual's body becomes saturated with the chemical signature of the plants it consumes. Some red cotton bugs feed primarily on cotton, building high levels of gossypol and related compounds. Others feed on alternative hosts, milkweeds, kapok, or various Malvaceae species, each carrying its own unique profile of defensive molecules. In effect, the bug's diet determines its toxic identity. Predators learn these differences. Birds that have sampled red cotton bugs on one plant species may avoid similarly colored insects on all plants. The risk is simply not worth the potential physiological distress. A single exposure can lead to minutes or hours of discomfort, during which the predator's ability to hunt is compromised. In ecosystems where survival depends on constant vigilance and energy efficiency, such distractions can have lasting consequences. Better to avoid bright red and black altogether. Within the bug, storage of toxins requires careful compartmentalization. Certain tissues accumulate the compounds directly. Others sequester them in small droplets that remain chemically inert until disturbed. The bug's circulatory system keeps these reservoirs isolated from vital organs, preventing self-poisoning. This delicate balance reflects millions of years of coevolution between insects and the chemical landscapes they inhabit. The bug must store enough toxin to deter predators, but not so much that its own biology becomes unstable. The warning colors serve as an honest advertisement of this internal chemistry. Unlike mimicry, where harmless insects copy the appearance of dangerous species, The red cotton bug truly carries the payload it displays. This honesty strengthens the signal across the ecosystem. Predators learn swiftly, and the pattern reinforces itself. A population of bugs feeding on highly toxic plants becomes a community of walking hazard signs, each one teaching predators to hesitate at the sight of red and black. Yet kleptotoxicity does more than protect individual insects. It shapes behavior, population structure, and even reproductive success. Bugs that feed on particularly toxic plants may have higher survival rates, but lower reproductive output due to the metabolic costs of detoxification. Others feeding on less toxic hosts may grow faster and reproduce more, but with greater predation risk. These trade-offs create diversity within populations, influencing how insects interact with plants and predators over time. In the cotton fields, the red cotton bugs cluster in small groups, each one tapping into the chemical reservoirs hidden within the seeds. Their feeding patterns affect the plant's physiology, reducing seed viability and altering cotton production. This makes them significant agricultural pests, but the biology behind their pest status is rooted in the same survival strategies that protect them from predators. The very compounds that make cotton difficult for animals to eat become the foundation of the bug's chemical armor. The afternoon ebbs into evening, shadows stretch across the field, and the temperature softens. The red cotton bugs migrate toward sheltered spots, their bodies still glowing faintly with their distinctive colors. They settle beneath leaves, on stems, or within clusters of unopened bolls. The toxins gathered throughout the day remain locked within them, waiting to send the next warning to anything bold enough to take a bite. In the quiet that follows, the ecological relationships woven through the field become clearer. Cotton plants defend themselves with chemistry. Red cotton bugs pierce those defenses and appropriate them. Predators learn caution. Populations adjust. Each actor in this chemical drama influences the others, forming a network of interactions that determine who survives, who adapts, and who retreats. The red cotton bug's body becomes a capsule of stolen toxins, bright, bold, and unapologetically dangerous. It carries the warning of the plant in its own skin. And in a world where predators must learn fast or suffer the consequences, that message is one of the most powerful defenses evolution can offer. On a warm spring morning, sunlight spills across an old courtyard, where cracked pavement meets the base of ancient lime trees. The air is still, heavy with the earthy scent of last year's fallen seeds and the faint sweetness of new blossoms. What at first appears to be a scattering of bright petals flickering in the sun is, upon closer inspection, something very different. A dense gathering of firebugs. Their bodies glow with bold red and black geometry, triangles, dots, and lines arranged in unmistakable warning patterns. Dozens of them. Then hundreds. Then more still, forming shifting carpets of color that move like slow, living embers spreading across the ground. A single firebug is striking. A cluster becomes a biological announcement. The coloration is not decorative. It is a threat broadcast in a language nearly all predators recognize. These insects are built as chemical fortresses, their bodies loaded with compounds designed to taste foul, disrupt digestion, and irritate tissues. But the true power of their defense comes not from the chemistry alone. It comes from the crowd. A blackbird lands nearby, hopping across the courtyard with its head tilted in curiosity. It approaches the firebugs cautiously. The insects do not flee. They do not scatter. Their stillness is not submission, but confidence. Bright aposematic colors are evolution's way of saying, try it and regret it. But every predator must learn this lesson at least once, and experience is often the most effective teacher. The bird selects the nearest individual, grabs it briefly in its beak, and then drops it almost immediately. The reaction is instantaneous. The firebug's body ruptures slightly under pressure, releasing a burst of dense, acrid-tasting fluid. The compounds saturate the bird's tongue with bitterness and irritate the sensitive tissues along the inner beak. Within seconds, the bird scrapes its beak against the pavement in an attempt to rid itself of the taste. It shakes its head repeatedly, chest feathers ruffling in discomfort. The firebug lies on its back for a moment, legs twitching, but even damaged, it has delivered its message. This red and black pattern is trouble. Nearby firebugs remain in place, undisturbed by the brief violence. They need no movement or aggression to defend themselves. One sampling attempt by a predator marks the entire cluster as unsuitable prey. In ecological terms, this is crowd psychology shaped by natural selection. The more individuals display the same warning signal, the more potent and memorable the lesson becomes. Predators avoid the pattern, not the individual. Thus, the survival of the group reinforces the survival of each member, and the vibrant aggregation becomes a collective shield, more powerful than any lone insect could muster. To understand why the bird reacted so strongly, one must look inside the firebug's body. These insects are specialists in chemical storage, feeding on seeds rich in oils, secondary plant compounds, and natural toxins. Some of these chemicals are metabolized. Others are absorbed almost unchanged, held within specialized tissues or stored in compartments where they can be deployed defensively when the insect is crushed or threatened. The compounds vary by plant species, but many share similar properties: bitterness, toxicity, and the ability to interfere with cellular membranes. Some firebug chemicals disrupt the lipid layers that surround cells, increasing permeability and causing them to leak. In small predators, exposure to these compounds can lead to digestive irritation, compromised gut lining, or even partial paralysis if enough toxin is consumed. Other substances interfere with metabolic pathways, forcing the predator's body to divert energy toward detoxification. These effects are rarely lethal to vertebrates, but they are deeply unpleasant. A predator that experiences this once rarely attempts to eat a firebug again. For invertebrate predators, the consequences can be more severe. Spiders that mistakenly capture firebugs often drop them quickly, instinctively avoiding the chemicals released as the bugs struggle. If a spider consumes even part of a firebug, the toxins can disrupt the spider's ability to digest prey properly or impair its coordination. Ants that attempt to dismember a firebug frequently abandon the effort within seconds, their mandibles recoiling from the irritants coating the bug's exterior. In this way, the firebug's defense is not a weapon, but a deterrent, a biological advertisement of unpleasant consequences. The insects do not need to run. They need only to exist in numbers, wearing their colors openly, letting one or two unlucky volunteers teach every predator within sight. The crowding behavior itself plays a key role. Firebugs are drawn to one another and gather in clusters across sun-warmed surfaces. Scientists believe this grouping serves multiple functions. Thermoregulation is one. By huddling together, the bugs absorb heat more efficiently, allowing them to become active earlier in the morning. Social and reproductive communication is another, as individuals release pheromones that attract others to safe or resource-rich sites. But perhaps the most profound function is the amplification of their defensive message. A single bright insect can be overlooked. 100 forming a crucible of red and black armor cannot. The sidewalk becomes a field of warning symbols, each one echoing the same evolutionary truth. Predators that ignore these signs suffer for it. As the day warms, the firebugs shift and reposition themselves. Some climb higher into the bark of the lime trees, gathering along crevices where seeds accumulate. Others spread outward in small exploratory movements, probing the edges of the courtyard. Wherever they gather, their bodies remain vivid. The grouping grows and contracts like a living organism, pulsing with slow, coordinated motion. Inside each firebug, the chemical factory continues its work. Toxins are processed, stored, and occasionally excreted in tiny amounts to maintain balance. These compounds are not produced in the same dramatic bursts seen in stink bugs or certain beetles. Instead, they accumulate over time as a consequence of diet and internal metabolism. The insect's bright coloration ensures these toxins can do their work long before they are ever released. A predator sees the warning and remembers, sparing the firebugs the need for confrontation. Still, the chemical payload remains potent enough to serve as a final line of defense. If a predator ignores the signals, the foul taste and cellular disruption act quickly, forcing the predator to retreat. The firebug may sustain minor damage, But the message is delivered, and the group survives. The cost paid by one strengthens the safety of the many. From an ecological perspective, these aggregations influence predator behavior across entire habitats. Birds alter their foraging routes. Small mammals modify their diet choices. Even invertebrates adjust their hunting patterns. The firebugs, by sheer presence and chemistry, shape the flow of energy through the local food web. They are small, yes, but their collective impact on predator learning and avoidance ripples through the ecosystem. As evening approaches, shadows stretch across the courtyard, and the firebugs begin to settle into cracks, leaf litter, and warm patches of bark. The cluster dissolves slightly, but remains dense enough to maintain its warning effect. Their bodies cool, their movements slow, but their colors remain as bold as ever, advertising the same uncompromising message, even in fading light. The nightmare scenario for a passing animal is simple. A seemingly harmless walkway becomes a living minefield of chemical defenses. The bright red shapes scattered across the cement are not decorations. They are miniature toxin reservoirs, each one capable of turning a casual bite into an unforgettable mistake. The safety they enjoy is not built individually, but collectively, an evolutionary triumph forged through crowd behavior and shared chemical armor. The firebugs rest as the last light fades, their red patterns glowing softly in the dusk. The courtyard grows still once more, but the warning remains etched into any predator that dared to test them earlier in the day. Late summer heat presses over the forest like a heavy, unmoving lid. Cicadas, sensing the deep warmth that softens bark and loosens sap, emerge in waves. Their bodies shimmer with a faint metallic sheen, wings trembling before they unfold with papery creaks. When they take flight, their calls rise into the canopy in a rolling chorus, an acoustic tide that swells and recedes, echoing from trunk to trunk. The forest hums with their presence, a sound so constant that silence itself becomes unimaginable. They are everywhere, clinging to leaves, clustering on branches, rising in sudden gyres of flight, and falling in soft glides toward the ground. For a few short weeks, the cicadas dominate the sensory landscape. But beneath the sound, another system operates, quieter, darker, and built on the chemistry of paralysis. Cicadas are not venomous creatures. They rely on speed, camouflage, and overwhelming numbers rather than toxins. Yet their bodies often become the vessels through which venom continues its work long after the sting. In this final twist of ecological design, cicadas become the living archives of chemical warfare, carrying the effects of venom into hidden corners of the food web. A female cicada lands on the trunk of an oak, its claws gripping the rough bark as it begins a slow ascent. The insect pauses frequently, shifting its weight and lifting its wings to better dissipate heat. The forest floor, still bright beneath the canopy, reflects a chorus of shadows across the tree. Beneath these shadows, a hunter emerges. A solitary wasp, slender and focused, scans the air for movement. Its antennae sweep back and forth, tasting the air for chemical trails. The cicada's vibrations, tiny tremors through the bark, give it away. The wasp launches upward with decisive clarity. It lands on the cicada's back before the insect can react. The cicada flares its wings, beginning a frantic buzz, but the wasp's legs hold tight. Its abdomen curls underneath, and with one precise motion, the stinger punctures the cicada's thorax. The injection that follows is not a deadly dose, but a neuromuscular override, a venom designed to collapse voluntary movement without stopping fundamental life support. Sodium channels lock open, muscles fire uncontrollably. The cicada shudders, its legs scraping wildly across the bark before stiffening. As the venom spreads, the cacophony inside the cicada's body changes. The muscles that once powered constant motion seize and then soften into a limp stillness. The respiratory spiracles along its abdomen pulse irregularly as neural messages become garbled fragments. The animal remains alive, fully oxygenated, with a beating heart and functioning organs, but its autonomy is stripped away. The wasp checks its work by gently pulling one wing. No resistance. The paralysis is complete. The cicada loses hold of the tree and drifts downward in a controlled fall, guided by the wasp's grip. The predator's legs clamp the cicada's thorax, and the wasp begins a laborious drag across the forest floor toward a small burrow hidden under leaf litter. The cicada's wings flutter weakly in reflex, but the motions are hollow, stripped of purpose by the venom's chemical command. Once inside the burrow, the wasp positions the cicada carefully, lays an egg, and seals the chamber with debris. The cicada, still breathing, becomes a living incubator. For the larva that hatches days later, this paralyzed cicada is not merely food, but an entire controlled environment. The venom ensures the host remains fresh. The tissues stay oxygen-rich. Decomposition is slowed to a crawl. The larva bites into the cicada's abdomen, sipping hemolymph while avoiding vital organs until the final stages of consumption. The host remains alive for much of this process, its internal rhythms continuing long after its nervous system has been hijacked. The venom echoes through this slow death, dictating not just the initial capture, but the entire arc of the larva's development. Elsewhere in the forest, another cicada lands in the web of a waiting spider. The silk trembles violently as the insect struggles, its wings cutting arcs of motion through the air. The spider approaches, legs lifting and lowering with ghost-like precision. Its fangs sink into the cicada's thorax, delivering a venom engineered for total shutdown. Unlike the wasp's targeted neurological compromise, the spider's cocktail includes digestive enzymes that begin breaking down tissues even as the cicada continues to breathe. The wings slow, the legs stiffen. A faint tremor ripples along the abdomen before the body softens. The spider steps back, letting the venom work invisibly within the cicada's internal systems. Proteins unfurl. Membranes weaken. The insect's tissues lose structure while the external form remains intact. The cicada hangs suspended in the web, neither dead nor fully alive, caught between states as the spider's silk sheath begins to envelop it. Over hours, the transformation continues. The cicada becomes a nutrient reservoir, its internal architecture liquefying into a dish the spider can later siphon with ease. Whether captured by wasp or spider, cicadas become repositories for venom's continued influence. Their bodies act as storage containers, preserving the venom's effects long after the initial sting. The chemicals that entered their bloodstream shape their ultimate fate, turning them into vessels of slow consumption. Venom lingers, reshaping tissues, altering neural signals, and creating an environment in which predators or their offspring can feed efficiently, safely, and for extended periods. The echoes of venom ripple further outward. Small mammals that stumble upon half-consumed cicadas may ingest tissues still laden with residual compounds, altering their digestion or behavior. Ant scavengers picking apart cicada remains can experience localized paralysis or digestive irritation if they consume areas rich in venom-altered fluids. Even fungi that colonize abandoned wasp chambers encounter tissues chemically transformed by the venom, altering their growth patterns. The compounds may lose potency over time, but the structural changes they cause persist, influencing every organism that interacts with the cicada's body. As one cicada becomes a silent vessel for venom, the rest of the forest continues its buzzing chorus. Sound waves roll across the landscape, rising and falling like an ancient song carried through the trees. The calls mask countless small dramas playing out under bark, beneath leaves, and within hidden burrows. Cicadas live short, intense lives, emerging in overwhelming numbers to ensure the species persists. Yet their individual fates often become entangled in the chemical strategies of predators. This final chapter draws the camera back, widening the lens to reveal a deeper truth. Venom does not disappear when the sting is delivered. It moves through the ecosystem, reshaping bodies, influencing feeding patterns, and altering the flow of energy. A cicada taken by a wasp becomes a cradle for new life. A cicada taken by a spider becomes a suspended reservoir of nutrients. In both cases, venom is the unseen architect, dictating how the body transitions from vibrant life to immobilized resource. In the end, the forest absorbs these transformations silently. The cicada's wings, once vibrating with summer's pulse, lie still as larvae feed or spiders sip, each movement a continuation of the venom's command. And when the chorus finally fades with the season's change, the echoes of venom remain, woven into the fibers of the food web, lingering in bodies turned into haunted meat, shaping the lives that depend on the deaths it orchestrates.